Hi, this is Dr. Judith Bailey here, and in this video we're doing topic 2.9, which is about photosynthesis. Here are some of the understandings that we have to go through. The first thing, we need to know that photosynthesis is a production of a carbon compound. In this case, we're going to see that it's glucose, but you can only do that by using light energy. We're going to talk about light energy. There's different ranges of colors, right? Going all the way from violet to red. We're also going to talk about pigments such as chlorophyll and how it's absorbing all colors except the greens and the yellow sometimes. Moreover, we're going to talk about how oxygen is a product of photosynthesis, and it comes out from the photolysis of water. We're also going to talk about how energy is needed in order to produce the carbs, right? Without energy, you cannot produce carbohydrates, and we also need carbon dioxide in order to produce a glucose. And after you know the whole thing about photosynthesis, we're going to see different factors that can actually cause photosynthesis to go faster or slower, such as the temperature, light, and carbon dioxide. We're going to look at these three factors and how they manipulate photosynthesis. We're also going to talk about the changes of the Earth atmosphere, ocean, and rock deposition due to photosynthesis throughout life. You should be able to draw, for example, the action spectrum as well as the absorption spectrum. These two are very important how to draw them, and I'm going to go through them with you. I'm going to talk about how to be able to design a lab in which you are testing the limiting factors such as the temperature, oxygen, sunlight on photosynthesis. And then here it says practical 4, which like I always keep repeating in these videos, in which the SL has 6 and while HL has 7. And that means these are practicals that falls on paper 3. So you have to know really well how to conduct them and know what they're all about. So in this case, we're doing about chromatography, in which chromatography is all about separating pigments. So we're going to separate different pigments in the leaf. So we're going to go through that. So let's start with photosynthesis. Photosynthesis, the overall picture, what it is. We all know that photosynthesis is happening in plants. And we all know that it starts with the sun right here, in which is sending its energy that will absorb by the plant. And then it will be converted eventually to glucose. So it's going from a light energy to become a chemical energy. So but besides that, what are the things that we need? We do need carbon dioxide. So that means the plant is absorbing carbon dioxide. The plant is also absorbing water. So we do need carbon dioxide and water for the plant. Now, the chlorophyll that is found inside the plant is absorbing that sunlight. Now, what does the chlorophyll does? It does pick up the energy of the sun, the carbon dioxide, and the water. And with the combination of these three, he will produce a glucose. And he will release oxygen as a byproduct. So that's the overall process of photosynthesis. Again, we're looking at the reactants. The reactants here is carbon dioxide and water. With the help of energy, as a product, we're going to produce oxygen and glucose. So this is the equation that came out from this. We have carbon dioxide, as you can see here. We have water. We have the light that is coming from the sun and the chlorophyll that is inside the plant. And as a conclusion to this, we have a product which you are producing glucose, oxygen, and water. So that's the full equation of photosynthesis. Now, here we can say that you have the light that is coming, and in the end is going to turn into chemical energy. Now, who is the chemical energy? It is the bonds found in the glucose molecule. So your photosynthesis is all about turning the light energy into chemical energy. And the chemical energy in this case, it is glucose, which again, glucose, another word for glucose is organic molecule. So whenever they say organic molecule, they're basically saying glucose, okay? So keep that in mind. Now, this is the definition as I was saying, but this is written, so for those who are better in visually seeing the words. So photosynthesis is the process of converting light energy into food energy, which basically is a chemical energy. And where is that chemical found? In the bonds of the glucose. So that is a perfect definition for photosynthesis. Now, photosynthesis is using the light, right? The light is coming from the sun, and then it creates chemical energy, in this case, ATP. Now, I'm going to explain more about ATP, although you should already know ATP from cell respiration on 2.8. So what is this energy being done? This energy that is being created, it can be used to make more food, such as glucose, or it can be used by the plant because plant also move. Plant also has chemical reactions. Plant also build stuff. So whenever they're moving, they're building or saying things, they need energy. So when the plant is taking the energy of the sun, right, the sunlight, it's not only making glucose so we can eat, it is also making energy for himself, for the plant itself, in order to be able to do its work. Okay? So once again, the light energy is converted to chemical energy. It's very important that these two words here you take into consideration whenever you're looking to multiple choice, whenever you're looking to write an answer. It's always going from light energy to chemical. Now, here's the relationship between photosynthesis and cell respiration. If you already watched 2.8 video, which is about cell respiration, and you come into this one, it will make a lot more sense to you. So right here, we have the two process, photosynthesis, cell respirations, and see how they're really interconnected with each other. Like, remember, plants give us the oxygen and we give the CO2 to the plant, which is clearly here. Now, the plant has the chloroplast, as you can see here, and the chloroplast is the one that is doing the photosynthesis. And what does that photosynthesis give us? It gives oxygen, and you see how here is written organic compounds? But organic compounds is the same thing as saying glucose. But so that word glucose and organic compounds will always be interchanged in the photosynthesis in questions. So do not panic if they write the word organic compounds in a test. It basically means glucose, okay? So photosynthesis is giving oxygen organic, which is given to us, and then our mitochondria does what? It breaks down the glucose and at the same time with the help of oxygen. And what we get out of that? Cellular energy, the energy for our cells to move. But while we're breaking glucose and oxygen here, what are we releasing? We're releasing water and CO2. What does the water CO2 do? They go back to the plant, and the plant is using it to go to photosynthesis. But the plant, don't forget that the plant starts with using the light sunlight, right? So this is combining both cell respiration and photosynthesis together. You can see here an overall, which is an interchange between us and the plants, and it's all between the photosynthesis and cell respiration. Now, since we're talking about sun and sunlight, let's look at what we can actually see and what we cannot. So here are all the sun rays. That means all the energy that has come from the sunlight. The ones that we can actually see is right here. This we can actually see through our eyes. We can see these different colors. However, what you see on the sides, the infrared and the ultraviolet, we cannot see their colors or see their shapes, but we can feel it. We can feel it through our skins, but it's not visible to us, okay? So, and you can see from this that the numbers that the lights that we can majorly see is between 400 and 700, which is very important for you to know. Now, these are wavelengths. When I say wavelengths, we're talking about energy in basically waves. And the nanometers right here is basically the unit that they use for the wavelengths. Now we're going to talk more about these visible lights, and we're going to see the different wavelengths of different colors and how interesting these stuff are. So right here, we have the sunlight, and we're showing different types of waves. We have short wavelengths, we have medium wavelengths, and we have long wavelengths. So if you are short, what short will be is that here the distance between this guy for the next wavelength that will be next to him, which we will show in the next slides, it is extremely short. So usually this one, the short ones are always blue. But if it's a long one, as you can see, the long one like this, it will be red. And green usually would have been in the middle. So you can see here is the green. So each one of them have a certain speed. That means that each one has a certain energy. So if you look at this and I ask you which one of these three have the highest energy, obviously the one that is short has the highest energy, which is blue. Blue here has the biggest energy. Which one have the least energy? It will be the red. Okay, and green will be in between. So the bigger the wavelength, the less energy it is. The shorter the length, the more energy it has. Why am I saying this? Because you can look here in the pictures. See, here are the different colors. And you can see the zigzag, like the mountains of each one, their wavelengths, right? And you can see that, for example, the violet, look at here, I'm going to highlight this with the yellow so you can see. Its zigzag is a little bit more faster, right? While with the red, look how it takes a little bit more longer time to do its little mountain curves, right? So what does that mean? That means here on the violet, it's going faster, the speed, than this one. Now, what exactly are these lines? If you really zoom in like really zoom in, okay? You see lines, like you see a line, right? You see a line, but if you really zoom in, it's actually an electron that is moving like that. And when he's moving like that, what is he doing? He's creating what we say a line here. Look, he's creating that line. That's what he's doing. Now, this electron, right, that I'm showing here, he has energy. So depending on the energy that this electron, and let me be here, this electron, depending on the energy of this electron, how much energy he has, this is how much you know how fast it's going to go or how slow it goes. So clearly, you can see that the electrons of the violets have a lot more energy because it just goes zigzag, right? And then the red, the electron of it is less, so you can see it goes slower, in a slower pace. So who has more energy giving out here? Usually, we were talking about the blue, indigo, and violet. These ones have more energy. That means they have more frequency, and that means their wavelengths are shorter, as you can see here. Now, if you want to know more about wavelengths, even though you don't need to know in detail, wavelength is just the distance between one tip of a mountain to the next one. That's what the wavelength is. So a tip from here to here, and then here is a tip to the next tip. So you can see the red has a bigger wavelength, right? You can see just from, look at comparison between this guy to this guy. You can see that this is smaller and this is bigger, right? This is big here, and this is small. So the smaller the wavelength, the faster they are going. It's like, think of this. Wait, let me put in a red, yellow color, white color. So imagine something goes like that. That means it's going really fast. But if it's going like, ooh, that means it's going really slow. So obviously, this guy right here, he has a lot of energy, and this one has a less energy. Now, this represents more like the red, and this one represents more like the violet. So violet has a higher frequency, has a short wavelength, but is very fast. It has a lot of energy, while red has a very low energy and is very slow. So this is how interesting it is when you're talking about colors, because usually depending on the color that you have, it can also influence you. So for example, just as an idea to give it to you, although you don't have to know this for IB, is that why do you think McDonald's, for instance, they use the red and the orange? See, they have less energy, right? So it puts the colors in the McDonald's to look like more calm and more relaxing, so you're eating, right? That's how they do it. And at the same time, it causes it to have less weight of heat on you. This is one of the reasons why we use those colors. But anyways, let's move along. So now we have to talk about pigments since we talk about the sun. So imagine this right here. This is the sunlight, right? This is coming from the sun, a white light. Now, if you knew about this prism, right? This prism right here, it's basically a crystal. And this crystal, when the light hits on it, it reflects all the colors. That's why you see right here the entire rainbow. What does that mean? That means that this prism right here, it does not absorb any of the energy. So it doesn't absorb any of that. Now, think for a second. If this little crystal right here did actually absorb all of this energy, you know what would happen to the crystal? It would crack. It would break into pieces. Why? Because it have absorbed so much sunlight. That means absorbed so much energy that the crystal will break. But this white crystal, what it actually does is it does not absorb any of this light energy. It just reflects them all. And that's why it just separates them into these colors. It separates the different wavelengths, the different energies. That's why you see the rainbow coming out of it. So the prism is another way of analogy towards the pigments that we find in plants. And we're going to talk about the different pigments in plants. So, for example, in plants, they have a pigment known as chlorophyll. Okay, that's the name of the pigment in the plants. Now, the chlorophyll, it absorbs all the energy of a sunlight. So imagine the sun is sending, right here, as the sun is going, sending that white light that I showed you right here, right? That's white light here. Let me erase this off. So imagine I'm talking about this white light. So if it's absorbing this light, right? So it's coming from the sun. Of course, that light is all the colors, right? What it does is it absorbs the blue and it absorbs the red. But this pigment does not absorb the energy of the green. And because it does not absorb the energy of the green is reflected. So that means the color green it hits on the leaf and then it leaves. And that's the reason why we see green leaves. Like if the leaf, the pigment, chlorophyll, did not absorb red or blue, then you would have seen a blue leaf or you would have seen a red leaf. But it actually absorbs the red and the blue energy, and it only reflects away the green energy, and the green energy is the one that you see. So chlorophyll is one of the pigments. There's so many other pigments and in the plants, depending on the pigment, the plant, of course. Now, where do you find this chlorophyll? Now, they're found, we know that plants have chloroplasts, right? And they usually are found here in these thylakoids. More closely, you can see the thylakoids here, right? Each one of these stacks are basically a thylakoid. And if you go inside of them, you will see a photosystem here, which you have all these pigments, or which are chlorophyll. And if you zoom it in, this is what it looks like, the structure of a chlorophyll. Do you have to know the structure and draw it? No, you don't have to. But you just can look at it. You see there's a ring here, and then there's a tail. That is the structure of a chlorophyll, but you do not need to know any of the details of that. You just need to know that chlorophyll is a pigment that is found inside the plant, specifically where? At the thylakoids, these thylakoids right here. So let me draw the chloroplast just to make sure you're okay with what I just said right now about the pigment. So right here we have the outer membrane of the chloroplast, right? So I'm going to write here the outer membrane. And then you have the second line here, which in this case, this would have been the inner membrane of the chloroplast. Because remember, chloroplast is a double membrane. And then you have the space here where you start making little stacks of coins like this. And then you just keep making more stacks of them, and you keep making another extra stack and just line up. Now, you can do this throughout the entire thing, but I just did some. Now, what are each one of these? Each one of these are known as thylakoid. Okay? The space here inside is known as stroma. Now, these here are known as lamella, which they are the ones that holds the granum together. What I mean by granum? This, a stack of bunch of thylakoid is known as grana. Okay, let me put it in a better place. Hold on. Let me line up here outside, and let's put here grana. So that's what grana is by you having a stack of thylakoids. So in each of these thylakoids that you have here, let me put green dots. Inside the thylakoids, this is where you have these dots. These dots that I'm putting here, these is where you find the pigment chlorophyll. So if I had to label here, chlorophyll. And remember, chlorophyll is not the same as chloroplast. Chlorophyll is the pigment. This whole thing is a chloroplast. So this is a picture of the chloroplast, and you should be very, very aware of how to draw the chloroplast and be labeling it all, okay? Because I'm going to be talking about photosynthesis, and photosynthesis has two processes. One of them is light-dependent reaction and light-independent reaction. So if I put it here in red, the first process is always happening at the colloid. So I put 1, 1, 1. Every one that I'm putting here, it means the first process. This is where the first process is happening. It is happening in the thylakoid. The second process of photosynthesis is happening in the stroma. So the 2, 2, 2 is happening here in the stroma. This is where the second process of photosynthesis. Now, that's very important for you to remember, okay? So the first process happens in the thylakoid. The second process happens in the stroma. This is just to give you a little bit of scoop of what I'm about to talk next. So I will talk about the two processes of photosynthesis. But for now, at least you have the structure, you have an idea where the pigment is located, which is the thylakoids of the chloroplasts. So let's talk about how many pigments there is. Now, usually there are about 40 to 50 chloroplasts in each plant cell. So we have trillions of plant cells, so you can imagine there's trillions of chloroplasts. And these chloroplasts have a zillions of pigments, right, that absorb a certain type of wavelength. In this case, they absorb all colors except green. And they are used, and when they absorb this energy, the energy that they absorb is used in photosynthesis. So you're using the energy of the red and you're using the energy of the blue, for instance, to make glucose. And of course, chlorophyll is the main pigment of all the plants. Now, how many thylakoids there is? Like in each thylakoid, there's about 200 to 400 chlorophyll pigments. Now, how many thylakoids? There's many thylakoids. So you can see it's a large amount of chlorophyll because there's a large amount of thylakoid and there's a large amount of chloroplast. So it's a lot of photosynthesis happening in a plant. Now, there's different types of pigments. We have chlorophyll A and B. This one, what it does is absorb all the colors except green. As you can see, that's why I even put in green here, because chlorophyll A and B does not absorb the green energy, but it does absorb the red, the orange, the yellow, the blue, the violet. Carotenoid, if you notice here, I put it in orange because it absorbs all the colors except red, orange, and yellow. So carotenoid is a pigment that does not absorb, or should I say it's reflecting the red, the orange, and yellow. So if you have carotenoid in a leaf, then that leaf will look like red, the leaf will look orange, or the leaf will look yellow. And then you have the phycobilins, and these guys basically reflect red. So if your leaf has the pigment phycobilins, that means it absorbs violets, blue, green, yellow, all of these guys, but it comes out red. So that's why sometimes in season you'll see the leaves of the color of the leaves changing in a plant is because one season there might be a lot of chlorophyll, and then maybe around spring they become red, is because now they produce more phycobilins. So they're always changing the pigments, amount of pigments in plants. That's why you see the plant changing from its leaf color depending on the season. That means depends how much it has a pigment in their leaf. So these are three pigments that you must know, especially chlorophyll and carotenoids. The first two, these two are the most important ones for IB. Now that you know about pigments and wavelengths and visible light and stuff, now there's two graphs that is extremely important that you know how to draw them, how to analyze them, and of course, be able to explain the difference between them. So right here we have two. The top one is talking about pigments and how pigments are absorbing different energy. The second one is talking about how fast photosynthesis is happening as the wavelength is there. So I'm going to go and talk about each one separately, and then I'm going to combine them again so you can have a better sense of this. Okay, but you can notice that both of them have exactly the same x-axis, which is the wavelength in nanometers. But their y-axis is completely different. As you can see, one is relative effectiveness of photosynthesis. That means how fast photosynthesis is happening. The second, this one right here, it's about how much absorption is happening. So let's look at each one more closely. The first one, it says absorption spectrum. That means you're absorbing something. In this case, we're talking about different pigments, and we're looking at each pigment, what is absorbing and what is not absorbing. That means what it's reflecting, okay? So in this picture, you can see I have 400, 500, 600, 700. These are nanometers. So these are known as the wavelength, as you can see. That's what I write in the x-axis. I put the wavelength with the nanometers. Make sure you put the unit nanometers. And then the y, we put the absorption of the pigments. So that's the x-axis. You have to be able to draw the x-axis. And then let's look more closely. Like this, let me take this out so it becomes more clear. So now let's look more closely on each of the pigments here. The first one that I'm going to check is chlorophyll A, right? Chlorophyll A is this one. It's the kind of green lighted. So between the 400 and 500, it kind of rises in the beginning. And you notice it doesn't start here on the y-axis. It starts a little bit up and it rises. That means here, what's happening? Let me erase this. I don't know why I did it. It just rises up like this. Means that here is absorbing what? It's absorbing mostly violet blue. But then it starts to drop as it gets down to 500. It stays low between 500 and 600. Why? Because here is usually green, because that's what's reflecting green. And then between 600 and 700, that's red. So I put red here, I put blue here. And then when it's red, what's happening? It's absorbing again a lot of red, and then it goes down. So this is showing chlorophyll A, what absorbs, what it does absorb. So if you look closely again, you will see that chlorophyll A absorbs a lot of blue, right? And a lot of red. That's why the mountains are like high. And then the one that it does not absorb, which is here, as you can see, it's low, it is the green. So it's showing what are the wavelengths that the chlorophyll A is absorbing and which ones it doesn't. Now we can do the same thing now for chlorophyll B. If you look for chlorophyll, which is the dark green here, it does a lot of absorption up here between 400 and 500, and you can see it is the highest peak, but then it drops between 500 and 600, and then it rises back up. So again, it absorbs a lot of blue-violet, but green, you can see that it's low, it's not absorbing. And then it absorbs red again. So once again, chlorophyll is a lot like chlorophyll A. So this is just showing how much is absorbing depending on the wavelength. And then we have the third one, which is the carotenoid. And you can see it starts at the point of the Y, and it's kind of slowly rising. It rises more towards 500, but then it drops here. So what does it mean? It does absorb blue, but when it comes to green, it reflects it, and it also reflects red out. So carotenoid is reflecting more the colors red, yellow, orange going out, but it absorbs the blue. So this graph right here is just showing the behavior of three pigments and which wavelengths that they absorb the most and which wavelengths they absorb the least. So it's all about absorption. On this one right here, it's talking about action. So the word action means there's a kind of process going. And this process going right now, it is the photosynthesis. That means how fast is photosynthesis happening? So you have the 400 right here, again the 500, the 600, and 700. If you notice, all of them is the same numbers that we have here, 400, 500, 600, and 700. So you put the same thing. And this one, also you have the wavelength, nanometers, right? But here on the y-axis, it's saying photosynthesis, while this one was saying just the absorption of the pigments. Okay, so that's the first thing that you can see the difference. So when it comes to the x-axis right here, it is the same thing for both, but when it comes to the y-axis here, it's different. Now this is just trying to put the three, what I'm doing is I'm taking these three pigments right here and I'm combining the absorptions of the energies that they picked up as well as the ones that reflected. So when you combine it, they do a big wave here because here is where they absorb most of the energy, but then all of them drop down here because most of here is doing what? They're reflecting it. And then again, here's mostly where they absorb again. So you can see that they mostly absorb what? Violet and blue, look at that, violet and blue, while here, green and yellow is where they mostly reflected. That means they're not absorbing this energy. And then from yellow on, such as orange, red, this is where they absorb again. So this is showing photosynthesis, right? But I'm not talking about the absorption. I'm just showing how it works like. So what is exactly this line trying to say? This line is not talking about absorption. This line is talking about photosynthesis. What I was telling you, yes, there was more absorption here, reflection here, more absorption here. But what does this has to do with photosynthesis? Well, think about it. If you absorb more sunlight, that means there's going to be more photosynthesis. So that means here you can see there's a large amount of photosynthesis happening. Here, because they reflect the light, that means there's not much photosynthesis happening. And then here, you can see there's a lot of absorption. That means, again, more sunlight absorption, more photosynthesis. That's why these two right here, a lot of photosynthesis is happening. So if you're absorbing red, violet and blue, or orange, red, a lot of photosynthesis is happening. If there's a yellow-green light, not much photosynthesis is happening. That's what the action spectrum is about. It's about how fast photosynthesis is happening. So it's kind of related to the other graph, but this is just talking about how fast photosynthesis is happening. So in this picture, I combine all together the two graphs. See, you can see in a more faded, that is showing chlorophyll A, chlorophyll B, and carotenoid, their pigments. And you can see in the bottom here, you have the 400 to the 700, and you have the wavelength light. Here we have both the absorption and the photosynthesis rate. So you can see that this line here, the darker one, that is showing the photosynthesis rate. And you can see that this line here, it is a combination of these three lines, as well as this one is a combination of this, and this line right here is a combination of these guys. So because we saw the behavior of the three pigments, we'll be able to figure out the action, the photosynthesis. So why you need to know this? Because in the test, what do? they will literally not give you anything. The only thing they will give you in a test, and this happened before, and I'm going to just highlight, they will give you this. They will make a line like this, and they tell you, draw the action spectrum. They will not give you the 400, 500s. They will not give you the y-axis. They will not show the lines. They would just give, literally, let me show you here writing. They would give you this. That's it. And you will have to add the 400 to the 500. You have to draw the graphs going like that. You have to put the y-axis. That means you have to build it from scratch. That is why it's extremely important that you are able to draw everything from scratch. Now, since you're drawing from the scratch, it's very important that you know how to spot them. Now, there's many ways to spot them. For example, you can see right here that the chlorophyll B is always a taller mountain than A and carotenoid. So A carotenoid, they're in the same level. And this is happening between 400 and 500, right? On between 500 and 700, you can see that B is smaller than the A. And then right here between 500 and 600, you can see that the three of them are low and carotenoid is already hitting the x-axis. Also, let's go more clearly when you want to look at the drawings, look at more. You see carotenoid, right, is touching the y-axis and is also touching the x-axis, it's mounted. However, chlorophyll A, it's above the B, but both of them are not touching the y-axis. And if you look at bottom here, they do touch at the end the x-axis. So these kind of little details, you need to be very careful and know how to draw. Now, you don't have to do an exact drawing like that, right? If you do a drawing like this, and then you have a drawing like that, and then a drawing like this, it looks fine because it's showing that B is bigger than the A in the carotenoid. So that's totally okay. As long as it looks very close to these drawings, then you're totally fine with it. Okay, so now that we got through this, now we're going to look at the two reactions. Because remember, if I told you, there's two processes that are happening in photosynthesis. There's the light-dependent reaction right here. That is the first process. And then the light-independent reaction. Now, the light-dependent, and you see the word dependent right here, it means that I am depending on the light to function. That means I need the sunlight. So it clearly says on the written word here, energy capturing reaction, which that means capture solar energy. So you do need the sunlight. And when you do get the sunlight, you convert it to ATP and ADPH. And this is happening as in the thylakoid. Like I mentioned, the first reaction is happening in the thylakoid. So the first reaction needs the sun. The second one, it says light independent, which that means I do not need the sun. I do not need the sunlight. In this one, you're taking ATP and ADPH, which are the products of the first one, and you're taking CO2 and you're creating glucose. And this is happening in the stroma, just like I showed you. Now, notice how here ATP and ADPH is the product of the first reaction, but here it is the reactant of the second reaction. And you can see that CO2 is only used on the second reaction, not on the first one. So we're going to go more in details for each one of them. So here we have the light-dependent reaction. Of course, the chlorophyll is absorbing the light, and then it's going to turn the light into chemical energy, which is ATP. Now, when it does this, it goes and breaks down water, which is known as photolysis. When you break down water, you create oxygen and hydrogen. Of course, oxygen is the oxygen you release, and the hydrogen you can use for the rest of the process. So then the hydrogen and the ATP will be used on the next process, which is light independent, and oxygen, as I said, was releasing. Now this was just a written. I'm actually going to draw it to make it more clear right here. So what is the process? It starts with you having the sunlight, right? So we're talking about, here we're talking about the light dependent. Light dependent is the first process. Now the light dependent location, where is it happening? It's happening in the thylakoid. Now, if you do the chloroplast very quickly here, right, you're doing the drawing, it's those stacks like that, the stacks that we had it, right? Those are the thylakoids. That's where you will find, let me put in green here. Actually, let's use this one. See, these guys right here, that's the thylakoid. So that's where the light-dependent reaction is happening. So when this is happening, what you have is if the sun is hitting its light on the plant, right? And the plant will use its chlorophyll and convert that sunlight right there into two things, ATP and an energy carrier, We call it an energy carrier because it's carrying energy. Oops. Which is known as NADPH. So he has energy on the hydrogen, he has energy on the phosphate. So these are two ways of carrying energy. Now, these energy are known as chemical energy. So once again, what did we do here? We took the sunlight and we converted it into ATP and ADPH. That was the whole light-dependent reaction. Now, the plant has the pigments, as we see here, right? Chlorophyll. But besides that, in order to do this whole process, it also needs water. So once the sunlight comes in, that sunlight hits the water and breaks the water into oxygen and hydrogens. Of course, there's two water here, H2O. Now, this sunlight, when it breaks water into forming 4 hydrogens and an oxygen, which this is the one that goes out of the plant. This process, you need to know this process. There's a name for this process, and this process is known as photolysis. Now, the word photo means light, and lysis means cut. So you're cutting with the light. So you can see that the sunlight right here, this guy is actually cutting what? It's cutting the water. So the sunlight is cutting the water and releasing oxygen as out. So every time we say the plant is releasing oxygen, it's actually coming from breaking the water inside of the plant. And that water is being broken down by who? By the sunlight that the plant picked up. Okay? So that is the light-dependent reaction. Very simple. Okay? So if you look at overall this, who is the reactants? If I look at the reactants here, the reactants is the sunlight and the water. Right? And who are the products? The products here, in this case, will be the oxygen, the ATP, and ADPH. Those are the 3 products. That's the process of light dependent. Now, this one can only happen during the day. Why is that? Because it depends on the light. It depends on the sun. So light dependent can never happen during the night. Then we go to the light independent. On this one, you see the ATP and the hydrogen, which came from the NADPH. Remember those 2 right here, right? We have this guy and this guy, these 2 right here. So these 2 that were made on the first process will be used to be working on the light-dependent reaction. So the hydrogen that came in there will combine with carbon dioxide. Now here's where you get the carbon dioxide, and you're going to form what? Organic compound. If you remember, I said organic compound is a glucose. And the ATP, what does it do? It just gives the energy, right? To do what? To continue with the process, with the continue building. When I say anabolic reaction, that means building up. What are you building? You're building the carbon molecules. Who are the carbon molecules? Glucose. So once again, ATP is giving its energy to build glucose. So that's what the light dependent is. It's about picking up those 2 molecules, again, as I was mentioning, ATP here and ADPH, and with the help of CO2 to create what? A glucose. So here is the second part. Let's look at the light independent reaction. In the light-dependent reaction, where is location? Location for this guy is in the stroma. So if I had to draw the chloroplast right here, and I have the double membrane, and I have the thylakoids, right? The thylakoids and the thylakoids. In this case, it will be happening here at the stroma, this area. So let me highlight here. All that's green area here, that's the stroma right there, the stroma, the stroma. So that's where it is happening. Now, light-dependent reaction actually has another name, which we call it the Kelvin cycle. It can also be known as the Kelvin cycle. Now, from the Kelvin cycle, what do we need? Now we need the CO2, we need the ATP, and the NADPH. Now, just to be clear, these 2 came from the first reaction. And CO2 came from what? Came from the air. So they come together, the 3 of them, and they're going to produce what? Glucose. Now, glucose, of course, it is the organic compound that you use to eat. No. So the whole point of photosynthesis was to produce glucose. So in the first process was just to convert the sunlight into ATP and ADH. And then the second process was to take ATP and ADH with CO2 to make glucose. That's what light dependence is about. Now, if you notice this one, we do not need the light. Although you do need the products from the previous process, you do not need the light. So this one can happen during the day, and this can happen during the night. So those are the two processes. So here is like a sum up of the two. You have the first process, which is the light dependent, and the second process, which is the light independent. In the first process, you have the sunlight and you hit the water. When you hit the water, what you did, you broke it down to hydrogen and oxygen. And what happens to oxygen? It just leaves the plant. Now, this part where the light hits the plant, it's called what? Photolysis, and you need to know this name. Now, what about the light that hits the chlorophyll? Well, that will turn into chemical energy. Who are they? The NADP, H, and ATP. Now, these two products right here, they move to the second process, which is the light dependent. So with ATP and ADPH and carbon dioxide, which is coming from here, where? From the outside into the plant, it causes a carbon fixation. Carbon fixation means the addition of carbon, which means you're putting a bunch of carbons together. That's what it means, carbon fixation, adding carbons together. And what do you get? Organic compound. In this case, who is the organic compound? That is glucose. So these are the two processes summarized very well by bioenergy in this picture here. It shows you how one process connects to the other one. Now, I can go through the process here and put all the reactants and products for each one because this is a good summary for you to understand what I mean by that. So here we have the equation, right? We have carbon dioxide, oops, I'm putting black, carbon dioxide, and we have water. That's the photosynthesis equation. And with that, you need light and chlorophyll. And what do you make in the end? You make glucose, some oxygen release, and some water, right? That's the full photosynthesis equation. But out of that equation, that's a summary of two reactions. And I'm going to show you here how each one is going this. So let's start with the light dependent, which is the first reaction. And if you remember this one is happening in the thylakoid, right? So this is a summary. So who are the reactants here? For sure, it's the sunlight, right? What else do you need here to start? You do not use CO2 on the first one, right? But we do use water. And what do we have here? Chlorophyll, if you want to put it. And you probably have NADP, and you have ADP, right? Oh, that look ugly. Let me put the A better here. Let's put here ADP and a phosphate loose. So these are the things that we need as reactants. Reactants means the first one to start the process. Then you have the product here. Now, what comes out of the product here is oxygen. And then you have ATP. Why? Because ATP was this combination of phosphate with ADP. You had NADPH. And that's it. These are the products that comes out of the light dependent. Now, where does the oxygen go? It goes out of the plant cell. Then we have the reactants. Now look at this. These two, which were products of the light dependent, they move to become the reactants of the next process, right? Of the light dependent, which is the second reaction, which is happening at the stroma. Now, these two are carrying energy, and they're the ones that are going to connect all the carbons. But where's the carbon? That's where it comes the CO2. That's CO2 right here. It comes on the second process, and you're going to join a bunch of like 6 CO2s all together. How you join them? Well, you use the energy of ATP and the energy of NADPH, and you're going to produce glucose. And this is how you separate. Now you can see that on the top, right here, let me put this in the, you can see in the top, this is like a summary. And when you see CO2 here, you think CO2 is the first product and the first reactant. No, CO2 is only used on the second part of the process. But here they're putting a summary. See, water here is the one that is being firstly used, right? And CO2 is only used on the second process. And then you can see glucose here is at the total end, while you can see the oxygen actually released on the first process. And even water here can be produced way later on the glucose. So the summary is on the top, but you can split the summary right here. And you can clearly look that you have the light dependent, which is the first reaction, and the light independent, which is the second reaction. And you can see here what are the reactants and products for the light dependent, what are the reactant products for this. But the most important part about this is that you have to see how they are linked to each other. You see, the product of the light dependent is actually the reactance of the light independent. So the light dependent here, this guy cannot function, it cannot work unless you go first through the light dependent. And the light dependent cannot start without the sunlight. So that's why they're all intercombined and they're all basically combined to each other. Okay? So now we understand the process of photosynthesis. Now we can talk about things that makes the photosynthesis go faster or slower. And that's what we call limiting. When we say limiting, that means they're somehow stopping. That means they can manipulate to go faster or slower. So let's look at some of these. Here are some examples that can cause the photosynthesis to go faster or slower, which it's the same thing as saying the word rate. So if you put more sunlight and if you put more CO2, great. In the beginning, yes, there's more CO2, more photosynthesis, or if there's more sunlight, more photosynthesis. But it's going to reach a point where it's going to plateau and there's not going to be more photosynthesis. And the reason why is because you have a certain amount of what? Of chlorophyll. Oops. You also have a certain amount of rubisco, which is also an enzyme that happens there. There's a certain amount of chloroplast. So just because you're adding more sunlight or more CO2, it doesn't mean that you're going to go faster. It's going to reach a point where if you only have 100 chlorophyll, no matter how much more sunlight you put, chlorophyll is going to say, hey, dude, we are occupied fully. There's no more we can do. You have to wait your line. That's why we hit the plateau. So this is what the graph will look like. In the beginning, yes, the more visible light, the more CO2, that means more photosynthesis for sure. But it's going to reach a point where it's just going to plateau. So you have to know how to do this graph. Remember that you always put the limiting factor on the x-axis and you put the rate of reaction always on the y. Then the other ones that affect this is amount of water, minerals, and temperature. Now, all of them, temperature, for example, yes, when you increase the temperature, there's more collisions, they're like excited, they're moving, they're working. And they keep working well until a point that they're going to hit the optimum, which often means the best moment in which everything is functioning really good. But after this, if you increase more temperature, what are you going to do? The plant is going to die. It's going to be burned. The enzymes are going to die. Everything's going to be nature, and it just drops. So usually in the test, you'll be drawing like this, and then it just drops, because that means that up to this thing here, the temperature. Yes, the more temperature, the more photosynthesis, or even the minerals, the more photosynthesis, even water. But imagine if you put too much water in the plant. We all know if you put too much water in the plant, what happens? The plant dies. That's why up to a point, yes, as increasing, yes, there's more water, yes, more photosynthesis, but after a certain point, now you're drowning the plant, so the plant will die. That's why these three, water, minerals, and temperatures, they're good in the beginning. They're increasing the photosynthesis rate. But up to a point, after that point, they all die, because then you are saturating, you're killing it with too much. So the next slide is just give you more in written words, which this is just to help you to write it down, because most of this I was saying in out loud. But here is again showing the graph of a temperature, right? There is a rise going on. Let me put in thing. There's a rise going on and there's a sudden drop. So the rise is because there's more kinetic energy, right? There is more action moving of the reactants. They're like speeding up, they're bouncing to each other. So there is a lot of photosynthesis happening until it reaches the optimal temperature, right? After that, what happens? Enzymes become unstable, denature, that means they die. And that's why the rate of photosynthesis drops so quickly. Carbon dioxide. Well, it's the same thing. In the beginning, you are rising the carbon dioxide, right? With more basically CO2 coming up. That means more rate of reaction, but eventually it will start to plateau. So what does that mean? Like, for example, oxygen is used up in the plant, is not in photosynthesis, it's only respiring. That's what it means, this part right here. Let me highlight in blue. Actually, let me erase this whole thing so you can see what I'm talking about. So basically what it's saying here, this area where it starts below zero, that's just when oxygen is used by the plant and there's no photosynthesis happen. After the zero, okay, I'm putting in yellow now, after the zero, now there's more CO2, and that means now the reaction started. Now, more CO2 means more photosynthesis, and it's true, more CO2, the more photosynthesis is happening. But it's going to reach to this part here where this is the maximum of photosynthesis can happen with the maximum of CO2 that can be used. After that, which is D, let me put it in yellow here, D, in this case, or should I say more E in this case, the range of values depends on the plant, right? But after this, it will be saturated, which that means it will not increase. It will just become a straight line. Let me put it in black here. The line will become just straight, and that means it has plateau. And this is the same thing with the light intensity. Light intensity, again, you just have the line here, which again, here is just oxygen being used. After this, more light, more photosynthesis, but then eventually will plateau. So if I had to draw this, I would actually put the light intensity here. And here I can put the rate of photosynthesis. So in the beginning, it's a curve, and then it just goes straight line. Same thing. After certain amount of light, you have a limited amount of chlorophyll, nothing can work afterwards. Now, we learned the 3 ways in which we can manipulate how fast the photosynthesis is. Now, we have to find experiments in which can actually measure these stuff. And there is direct and indirect way. And you have to know both ways because that's what I'd be asked of you. So the direct way, obviously, is oxygen, right? The more the plant is releasing oxygen, the more photosynthesis is happening, right? Because oxygen is a product. Also, one that can be directly and indirectly is CO2. The more CO2 is absorbed by the plant, that means more photosynthesis is happening. And then biomass here means the weight of the plant, right? Now this one is indirectly. That means if the plant is getting heavier, that means there must be some kind of production of more glucose, which that means more glucose, more weight. But it's not 100% accurate, so that's why we say indirect. So these are 3 ways in which you can measure photosynthesis. You check the oxygen produced, or the carbon dioxide that is being used, or the amount of weight of the plant. So let's look at examples for each one. So for example, we have here CO2, right? The uptake. There's so many ways you can do this. One of this is that you get a beaker, right? And in that beaker, you put a solution in which there is CO2 here, and you put a plant. I'm just going to put P as a plant, right? And then, of course, you block this and you allow the plant to go through the process of photosynthesis. So you measure it, let's say day 1, you measure it. What are you measuring? You're measuring the pH of the water, because remember, the more CO2 there is inside the water, what happens to the pH? The pH drops, so it's more acidic, right? So if the plant is taking CO2 inside of it and turning it to glucose, what's happening to the pH? The pH is rising, so the pH is becoming more basic. So in the beginning, the water might be acid, but then by the day 5, the water becomes basic. What does that mean? That means the CO2 was absorbed by the plant, so that means there is less CO2 on the water, and that means the water had become more neutral. So this is one way to measure to see if the CO2 has been absorbed by the plant. Another way is you put again now a plant on the box, and it's closed up, and you put the plant right here, and you have a certain amount of CO2 in the air, and you measure how much there is on day 1. Then you put a Vernier probe right here, like it's basically a stick here, and this probe is connected to a computer, and you measure the day 1. And then you go and measure on day, let's say 5, and you figure out day 1 what was the amount of CO2, and then what was the amount on day 5. And you can see how much CO2 has been absorbed by the plant. As you can see, if the CO2 dropped, let's say here was 100 and dropped to 50, that means there was 50 photosynthesis happening. So that means 50 of the CO2 was absorbed by the plant. So that means there was photosynthesis that was actually happening. Now, this is a Vernier probe. We can find this in your labs in some schools, but they actually just put the probes there and see how much the CO2 is reducing in the air around that plant. So that means photosynthesis is happening. When it comes to oxygen production, how that happens? Here's a perfect picture. You put a lamp, and this is algae. This is basically a water plant. And water plant, what does the plant do? Plant produces oxygen. What happens when you put oxygen in water? oxygen and water becomes bubbles. So every time a plant is releasing a bubble out, you count them. So you can say in 30 seconds, I want to see how many bubbles are coming out of that plant. And that's how you know how much the rate of reaction is, because if you see a lot of bubble coming out, that means there's a lot of photosynthesis happening. If you see little amount of bubbles coming out, that means the photosynthesis is going very slow. So that's one way. It's just counting how many bubbles are coming out of the plant. That's one way to count. Another way is just to get a box and you put the plant. Of course, you close the box and you put a Vernier probe here into the computer and you measure the oxygen in the atmosphere on day 1, and then on day 5 you measure again and you see it. So the expectation is that if you started with 650 oxygens in the first day, you should have like a 500 on the day 5. Why? Because the plant is producing the oxygen. But if you don't have 500 and you have only 20, what does that mean? The plant was not doing photosynthesis, because if there is no oxygen out, that means there is no production of photosynthesis and there's no oxygen release. So this is another way to measure oxygen. And then it comes to the mass, which that means the weight of the plant. How can you measure this? So what you do is you get yourself a scale, right? And you have the numbers here. And you put the plant here with the vase. And you have the plant here, blah, blah, blah. And you measure on day 1 how much that weight is. And then you count maybe after 10 days, you can go and do the same thing. You put the weight here, you put the plant, right? I'm not going to draw the same plate here. And you check the weight. So if the weight here, let's say the weight was 3 kilograms, and then after 10 days it was 5 kilograms, we are assuming that the extra 2 kilos that was produced was actually glucose. So we're saying that there was photosynthesis, that photosynthesis did happen and glucose was produced. The only problem with that, of it being indirect, is because what if from day 1, while you were going through the day 3, day 4, day 5, what if you were watering the plant? What if that 2 kilos, out of that 2 kilos, 1 kilogram is glucose and the other kilograms was just adding water to the plant every day to make sure the plant is growing? So it's an indirect. This one is not very accurate. So eventually it's not a good way to measure photosynthesis, because you don't know if those 2 kilos was all of it glucose made from photosynthesis, or out of those 2 kilos there was some of it for water, or there was a branch that was built, or the root got bigger without the glucose in there. So it's very indirect. So that was ways to measure photosynthesis rate through oxygen, CO2, and mass. Now, this is one of the labs that you need to know how to use it, which is known as chromatography. Now, chromatography lab, it's made of this paper right here. This paper here, it's actually made of chemicals. And what it does is these chemicals have charges, like plus is negative. So they basically move along according to the charges, right? Now, how does this process really work? The chromatography paper. So first thing we do is get a chromatography paper, and then we take a pencil and we make a line in the bottom here. So this will be the origin. Then we take a leaf and we place the leaf right here on top of the line, and then we rub the leaf on it. So when you're rubbing the leaf on it and then you take the leaf out, it will look like we have some leftovers of the pigment. So now you can see a smash of grain on that line. Then what's next thing to do? The next thing to do is put this thing on a cup. So I'm going to put it here. I'm going to put this thing in a cup. So now it's inside of a cup. And in the bottom of that cup, what I'm going to do is I'm going to use purple here. I'm going to put an alcohol. In this case, it can be acetone, right? So we put alcohol liquid right here. Now this alcohol has to touch at least the bottom of the chromatography paper. It doesn't have to touch all of it, just the bottom of it, okay? So when it touches the bottom of it, this is alcohol again. Now you know how water and liquids can go through the paper and then you see the paper absorbing that liquid? It's the same thing. The paper is absorbing that alcohol. So that alcohol, I don't know if you have different colors. Yes, it does. Okay, so the alcohol is kind of absorbing by the paper and it's moving up. It's moving up like that, right? So as it's moving up, through the paper, right? Because the paper is absorbing the alcohol, it's going to reach a point where it just stops. So this whole purple thing is alcohol passing through the paper and it reaches a stop. So this is the level here. This is the level in which the distance that the alcohol, travel. Now, you see the alcohol, they touched that green part. When it touched that green part, it eventually caused some pigment to separate. There was a yellow here, and then you might have a light green happen here, and then you might have a darker green happen here. Oops, it didn't change the color. Okay, here. Another green color here. It can even have red. And the red here. Let's say. So what happened here is this this one part here, pigment, right? This whole smash of the leaf eventually separated into 1, 2, 3, 4 pigments through the chromatography paper. And you can see it right here. Look at the picture. You can see the different colors being spread on that paper because the alcohol spread them and the chromatography helped separate these pigments. Now, each one has a certain distance, right? The distance from the origin to this thing, and the distance from the origin, and the distance from the origin, and the distance from the origin. So they all have their distance from the origin and the distance of this, of the alcohol traveled. From that, you can find the RF, which that means the value of the pigments. So with that, you can see the pictures here. Chromatography paper, you have the alcohol in the bottom, and then you can see the separations of the different pigments, how far they traveled. So carotene will always have a value of RF value of 9.95. And xanthophyll, this pigment has a 0.71. You don't have to memorize these names. It's just showing you how different pigments travel different distances. And how do I get these RF value? Well, I need to find the distance of the pigment to its origin. So whatever the distance is, and then I divide that by the distance of, not here. Let me erase this. I look at where the solvent ended right here, and I find the distance from the origin, and I will put here, I'll put X. So you divide the distance of the pigment travel by the distance that the alcohol travel, and you will get the RF value. Very straight. This is a lab that the teacher has to do it in class with you, so you can visually see it. But the whole purpose of this chromatography lab was just to separate the pigments. And you can do this with makeup. If you put, for example, lipsticks here, you smash it here, and then you're going to see the different colors of the lipstick coming out, the different pigments. So chromatography, it's all about separating pigments. Remember that. The purpose of chromatography is to separate the pigments found in plant leaves or found in a makeup or whatever, anything that has a mixture of colors. And the last thing you have to know for this topic is about how oxygen has been changing and the plants, how they have been reacting from the early days, doing photosynthesis in the atmosphere, in the oceans, and the rock. So you can see on stage 1 right here that in stage 1, there was no oxygen in the atmosphere or in the oceans. Then on stage 2, you start to see some oxygen being produced, but they're mostly absorbed by the ocean and some rock. On the stage 3, you start to see the oxygen turn into gas. That means you're starting to see the oxygen in the air more, right, as it coming out of the ocean. And then eventually on stage 4, that's where you see oxygen accumulate in the atmosphere, filling up not only the atmosphere, but around the oceans and everything. So what is this trying to show? It's just showing how photosynthesis, it kind of rose as you went through the 4 stages. So we are talking about years, right? Billions of years ago. We're talking about all this before it was really no oxygen, and with time, oxygen was starting to rise in the atmosphere. And since oxygen was starting to rise, we start to have more plants on the land. So you can see that from this graph here, that if I ask you, was there first algae or there was a plant in the land, you obviously know that since oxygen started in the ocean, that means most likely algae were the first photosynthesis to happen. That means photosynthesis happened first inside the ocean before the photosynthesis could actually happen outside of the ocean. And that is pretty much it for this topic. Here is the study guides. You can always go back to these questions in the video, as well as these questions, and that's pretty much it. Thank you.